Hello everyone, and welcome to episode 13. Today I'll be opening up a new mini-series exploring genes, genetics, and meiosis. This episode will begin the mini-series by exploring the experiments and research of the Austrian monk Gregor Johann Mendel. Between the years 1856 and 1863, Mendel conducted experiments with pea plants. These experiments involved the reproduction of successive generations, where the traits that emerged in the phenotype, or the outward appearance, were recorded and cross-referenced to their parents and grandparents. In this way, Mendel's work developed the rules we use for understanding and explaining heredity. Collectively, these rules are called the laws of Mendelian inheritance, honoring their creator, Gregor Johann Mendel. He announced his work to the public in 1865, but received little fanfare and attention for nearly 34 years. Unlike Darwin, his work was not enthusiastically appreciated by his contemporaries. It wasn't until later, around the 20th century, that experiments with more refined technology backed up almost all of Mendel's claims, giving his scientific literature validation as the brilliant work that it was. It was truly ahead of its time in many ways. So Johann Mendel was born to an Austrian family living in what's now known as the Czech Republic. His early life was on a farm, where he learned beekeeping and gardening. He studied at the University of Olomouc from 1840 to 1843, but suffered from both financial troubles and illnesses. In order to continue his studies without worrying about the expense, he joined the church and became a friar, and he received the name Gregor. For a time, he tried but failed to become a substitute teacher at the Augustinian St. Thomas's Abbey. Having then studied physics at the University of Vienna, he came back to try to become a teacher, but failed again. It was at the University of Olomouc, early in his scientific career, that Mendel conducted his famous experiments on plant genetics. He studied the pea, using it as a model organism due to its rapid growth cycle, abundance of seeds, and the minimal care that it required. He selected 7 traits about the pea plant to study, to track and observe across multiple generations. These traits were the plant's height, the shape and coat tint of its seeds, the shape and color of its pods, and the color and location of its flowers. The species of peas that Mendel was working with had both male and female parts, and would normally self-fertilize. Instead, Mendel removed the male parts from the plants before they produced and released pollen, in order to prevent self-fertilization. Mendel then took the pollen in the male parts, and brushed it onto the female parts of the other pea plants that he wanted to crossbreed with. This allowed him to control which plants mated with which, to select favorable or pure strains, and create populations with specific traits. Traits are the phenotypic expression of an allele of a gene. There's a few important terms in that sentence that I should define. A gene is a small region of a strand of DNA, which codes for a protein or a group of proteins integral to the expression of a particular trait. The trait is the actual physical characteristic, as part of the phenotype, or the exterior cosmetic and physiological appearance of the organism. Genes code for traits, and alleles are different versions of the gene that code for different versions of the trait. For example, everyone has a hair color and an eye color, which are both traits. And we all have genes for hair color and eye color, which code for these traits. Alleles code for the specific variations to that gene. So the genes for the eye color trait have multiple alleles, one coding for blue eyes, another for brown, for green, hazel, etc. Similarly, the genes for the hair color trait have multiple alleles, one coding for blonde hair, one for black hair, another for red hair, for brown hair, or some other intermediate color like strawberry blonde or ombre. Mendel's pea plants also had alleles for their traits. The first trait that he crossed was the shape of the pea seeds. Mendel took pea plants from 2 purebred populations, one that was homozygous for round seeds, and another that was homozygous for wrinkled seeds. I've just introduced a few more terms that I should explain and define. Purebred, or a pure genetic line, means the individual will produce another individual almost identical to itself if it's crossed with another individual from the purebred population. Basically, it's a stable population with a set group of characteristics, and so when you breed within the population, the offspring all have these characteristics as well. They're all purebred. In relation to dogs, the term mixed breed then refers to a dog whose parents are 2 different breeds of dogs that came together to create the mix. Homozygous means that the individual has the same allele for each copy of the gene. Remember that diploid species have cells with 2 copies of the genome. They thus have 2 copies of each gene. When each gene has the same allele, the individual is homozygous for that trait. They have two of the same alleles, one on each gene on each copy of the chromosome. By contrast, when each allele is different, the individual is heterozygous for that trait. So they have two copies of the gene, one on each copy of the chromosome, except they have two different alleles. So say for hair color, this is simplifying, but say for hair color, one allele is for red hair, and the other allele is for blonde hair. If the blonde hair allele was dominant, the offspring would have blonde hair. But say they were co-dominant, or they didn't, express a dominance over one another, then the child would likely have a mix of red and blonde hair, and their hair would be like a strawberry blonde or like a coppery brown. So Mendel crossed a purebred round seed pea plant with a purebred wrinkled seed pea plant. So this is a cross between round seeded peas and wrinkled seeded peas. The offspring of this cross, to much surprise, all had round seeds. This discovery was important because it meant the traits did not blend together into some kind of intermediate, as was predicted by the prevailing theories of the day. The discovery also posed a question: where did the wrinkles go? To find out, Mendel took the offspring generation, called the F1 generation, or just F1, and he had them self-pollinate themselves. You see, Mendel reasoned that some genetic determinant existed for both round and wrinkled seeds, and both determinants existed in the F1 generation, even though only the round trait was being expressed. Interestingly, the F2 generation, or the offspring of the self-pollinating F1 generation, they had both wrinkled and round seeds, but in a ratio of 1:3. This means that out of every 4 offspring, 1 had wrinkled seeds, while the rest had round seeds. The importance of this ratio will become apparent when I talk about the mathematics of heredity. Based on the results of both the parent crossing and the F1 self-pollinating, Mendel reasoned that some traits were recessive, while others were dominant. In the case of his peas, the wrinkled shape of seeds was recessive, while the round shape was dominant. It's important to understand that recessive and dominant in a genetic context does not imply that the dominant gene offers high fitness, or that the recessive gene lowers fitness. These terms, dominant and recessive, are only meant to be used in the context of which allele gets expressed in the phenotype of a heterozygous individual. As for the other genes in the pea plant that Mendel studied, they all had their own alleles as well. Seed color could be yellow or green. Yellow was dominant while green was recessive. The pod shape could be inflated or constricted, with the inflated shape being dominant and the constricted shape being recessive. A green pod color was dominant, while a yellow pod color was recessive. Purple flowers were dominant, while white flowers were recessive. The allele that positions flowers axially was dominant, while the allele that positioned flowers at the ends of branches was recessive. Lastly, it seemed that taller height was the dominant allele, whereas a dwarfish height in the pea plant was due to the recessive allele. How did Mendel explain the disappearance of the wrinkled seeds in the F1 generation, and their reappearance in a quarter of the F2 generation? His reasoning was ahead of its time. Mendel figured that each pair of genes must be separated into separate gametes. In this way, each gamete possesses one allele for each gene in the organism's genotype. As humans are diploid, we have 2 copies of each of our genes. When our chromosomes are divided during meiosis, 1 copy goes to each daughter cell, or each allele. Thus, each allele is haploid, in that it only possesses a single copy of the genotype. This concept is called the principle of segregation. So let's consider the first parent pea plants that Mendel used. These were purebred strains that varied in one characteristic: seed shape. Because round seeds are dominant, we can represent this allele with a capital R, call it big R. As wrinkled seeds are recessive, we can represent this allele with a lowercase r, called little r. So the parent which was purebred for round seeds can be described as having a genotype big R big R. They do not possess the allele for wrinkled seeds. The other parent is purebred for wrinkled seeds, and they can be described as having the genotype little r. Both big R and little r are homozygous genotypes. They possess the same allele in each copy of the gene. The F1 generation, however, possessed both a big R and a little r, which they got from their respective parents. Every F1 plant had both alleles for round and wrinkled seeds, big R, little r. But because big R is dominant, only it was expressed, and thus the seeds were only round. Mendel argued that each R was separated into individual gamete cells, alongside one copy of every other gene in the genome. So when the heterozygous F1 generation self-pollinated, Each egg could possess a little r or big R allele for the seed shape gene. Similarly, each sperm could possess either a little r or a big R allele for the seed shape gene, and could bind with any particular egg. This means the offspring of the heterozygous self-pollination would exhibit three combinations of alleles. The first is big R, and the second is little r, and these are both homozygous. The third combination is big R, and these are heterozygous offspring. The big R offspring will have round seeds, while the little r offspring will have wrinkled seeds. The heterozygous big R offspring are much like their F1 parents. They have both types of alleles, but only the dominant one is expressed, so these have round seeds. You can visualize this genetic crossing with a Punnett square. Imagine a square, and inside of the square are two crossing lines, splitting the square into four smaller boxes. The top of the square will represent one parent, let's say the mother, while the left side of the square will represent the other parent, the father. So above the top left box, write a big R. Above the top right box, write a little r. This represents the big R genotype of the mother. Now to the left of the top left box, write a big R. To the left of the bottom left box, write a little r. This represents the genotype of the father. So now we can cross these genotypes. The big R of the mother can be written in each box below it, specifically the left side, upper, and lower boxes. The little r of the mother can be written in each box below it, specifically the right side, upper, and lower boxes. The big R of the father can be written in the top right and left boxes, while the little r of the father can be written in the bottom right and left boxes. So what does this give us? Each box in the Punnett square has two letters, corresponding to the possible genotypes of the offspring. The top left square has two big R's, one each from the mother and father. This square represents 25% of the possible offspring, who will be homozygous for round seeds. The bottom right square has two little r's, again one each from the mother and the father. This square represents another 25% of the possible offspring, who will be homozygous for wrinkled seeds. Now take a look at both the upper right and lower left boxes. Each box has a big R, little r heterozygous genotype, and each box represents 25% of the possible offspring. These offspring will be heterozygous, but because big R is dominant, they'll express round seeds in their phenotype. So Mendel's F1 self-pollinating plants gave birth to 7,324 offspring. 5,474 of these offspring had round seeds, while 1,850 of them had wrinkled seeds. Mendel did not have access to technology that would have enabled him to examine the genes on the chromosomes themselves, but his experiment demonstrated the 3-to-1 ratio, where 25% of the offspring expressed the recessive trait, and 75% of the offspring expressed the dominant trait. This is a pretty clean and simple demonstration of how alleles are split up from parents to offspring. These experiments were a mono-hybrid cross, a cross performed between two parents who differ in only a single trait. In the case of Mendel's current experiments, the only differing trait was seed shape, wrinkled or round. So what would happen if you made a dihybrid cross, or a cross between parents who had two different traits instead of just one? Consider the seed-colored trait that Mendel studied. The dominant allele was for yellow-colored seeds, represented by big Y. The recessive allele was for green-colored seeds, represented by a little y. Now consider two hypothetical parent plants, one with round yellow seeds and the other with wrinkled green seeds. These parent plants make their dihybrid cross, producing an F1 generation of offspring that are all heterozygous for both traits. Each allele exists in each individual offspring, but the dominant alleles are being expressed. So in the case of our hypothetical dihybrid cross, the F1 offspring would have round yellow seeds, as these are the dominant alleles in their heterozygous genotype. The interesting stuff starts happening when the F1 generation is self-pollinated, or cross-pollinated with another member of the F1 population. The F1 offspring has alleles that can do two possible things. During meiosis, they can be assorted independently into the gametes, or they can be sorted depending on where one particular allele goes. The former possibility is called independent assortment, while the latter is called dependent assortment. If the alleles were to be organized according to dependent assortment, the alleles for seed shape would somehow be associated with the alleles for seed color, or vice versa, such that when one of these alleles was moved to a gamete, the other allele was brought with it. As a result, the offspring would all share the same groups of traits. The hypothetical dihybrid cross would produce results very similar to our monohybrid cross: 25% with all dominant alleles, 25% with all recessive alleles, and another 50% with heterozygous alleles for each trait. However, when Mendel made his dihybrid cross and self-pollinated the offspring to create an F2 generation, this is not what he found. What Mendel found instead was far more supportive of the concept of independent assortment. That is to say, during the formation of each genotype that would be given to each gamete, the alleles were sorted independently of one another. It did not matter where, for example, the big R went. The little r, big Y, and little y alleles are sorted independently of the big R. This is true for every allele. If we were to map this out with a Punnett square, it would need to have 16 boxes, 4 to a side. The tops of each column and the left sides of each row would be represented by one possible copy of the gametes they could receive from each parent. Like the first example, let's say the top represents the mother while the left side represents the father. Each in the F1 generation, being heterozygous for both traits, would give 1/4 of their gametes the big R, big Y genotype. Another 1/4 of the offspring would be given the big R, little y genotype, and another 1/4 would be given the little r, big Y genotype, with the last 1/4 being given the little r, little y genotype. So with each genotype written along the top and left side of the Punnett square, we can begin combining gametes to see the possible genotypes that could exist in the offspring. Mendel performed this exact experiment, getting 556 total offspring in his F2 generation. 315 of these offspring, or about 9/16 of the total, expressed the dominant alleles with round yellow seeds. 108 of these offspring, or about 3/16, had round seeds that were green in color. 101 of them, or close to another 3/16 of the offspring, had yellow color but wrinkled shape. These two groups of offspring are heterozygous for one gene, where the dominant allele is expressed, and they're homozygous recessive for the other gene, where the recessive allele is expressed. The final 1/16 of the offspring, about 32 individuals in his total population of 556, were homozygous recessive for both traits, expressing a phenotype with green color and a wrinkled shape. This 9:3 to 3:1 ratio in the dihybrid cross is like a mathematical square of the 3:1 ratio in the monohybrid cross. It also demonstrates that the principle of segregation in monohybrid crosses translates in dihybrid crosses into the principle of independent assortment. Mendel tested the principle of independent assortment in a different way, by using a test cross. A test cross involves a parent organism that's homozygous recessive for all alleles, and another parent with an unknown genotype. The offspring of such a test cross can reveal information about the parent with an unknown genotype. The parent with the homozygous recessive genotype will always pass on recessive alleles, as it's all they have. So any dominant alleles of the parent with an unknown genotype will likely be expressed in the offspring. Specifically, if the parent is heterozygous, it produces gametes with both dominant and recessive alleles. The offspring has a 50% chance of inheriting either one, depending on which allele is in the gamete that fertilizes the egg. If the parent is homozygous, it produces gametes that have the same allele for each gene. The offspring of a homozygous parent will always have one copy of the same allele, dominant or recessive, as there's no alternative allele available from that parent. Gregor Johann Mendel performed this experiment as well. Mendel's homozygous recessive parent had the genotype r, little y. It was homozygous recessive for both genes, and its phenotype was a green, wrinkled seed. The other parent Mendel used had an unknown genotype. However, he could observe the pea plant's phenotype, which expressed round yellow seeds. From these observations, Mendel reasoned that the unknown genotype must possess at least one big R and one big Y, out of 4 possible alleles. There were 2 possible outcomes to this test cross that Mendel created calculations for. The first was if the unknown genotype was homozygous dominant for both genes, in which case every offspring would receive one dominant allele from that parent and one recessive allele from the other parent. Every offspring would have the same genotype, with heterozygous alleles for both genes. The second possible outcome was if the parent's unknown genotype was heterozygous for both genes. In this case, the offspring population could be divided into quarters, one having round yellow seeds, another having round green seeds, the third having wrinkled yellow seeds, and the last having wrinkled green seeds. This represents every possible combination of alleles, equally represented in the offspring population. This demonstrates the validity of the principle of independent assortment. Recall that at the beginning of the episode, I mentioned how Mendel's work wasn't appreciated for its worth until the turn of the 20th century. The first work that validated Johann Mendel came in the form of a hypothesis published in 1902 about meiosis. Two scientists working independently of one another came up with a hypothesis that meiosis could explain both principles I discussed earlier, the principle of segregation that was demonstrated in the monohybrid cross, and the principle of independent assortment that was demonstrated in the dihybrid cross. I'll spend the entirety of the next episode talking about meiosis in detail, but for now, all you need to know is that meiosis is the process by which a diploid progenitor cell makes copies of its pairs of its chromosomes and then divides into 4 haploid gametes, with each gamete receiving 1 chromosome from the 2 copied pairs. These two scientists were named Theodore Boveri and Walter Sutton. They identified Mendel's so-called genetic determinants as specific loci on the chromosome. You see, the chromosome is a giant, coiling, piled-up mass of a DNA molecule, which is like a massive string. Portions of this string that code for proteins are called genes. The gene exists as a discrete stretch of the DNA string. This point on the DNA strand, as is any other specific point on the DNA strand, is called a locus, or a loci to be plural. In Mendel's experiments, it was the loci for the various traits, like round shape or green color, that were scattered between gametes during independent assortment. During independent assortment, it's assumed that genes are on different chromosomes. If genes are on the same chromosome, they would demonstrate some degree of linkage. Sister chromatids are pulled apart during a middle stage in meiosis, each pulled into the body cavity of one of the 2 emerging gamete cells. Genes on different chromosomes are pulled independently of each other, but genes on the same chromosome will generally go to the same gamete. There was a man named Thomas Hunt Morgan, an American scientist, an evolutionary biologist and geneticist who was active in the early 20th century, and he conducted research on, among other things, meiosis in a model organism called Drosophila melanogaster, aka the common fruit fly. One of Morgan's experiments with the Drosophila looked at the linkage of genes on the same chromosomes, and how they moved during meiosis. His experiment looked at 2 genes on the creature's X chromosome. One of these genes, W, was for an eye color trait, and another gene, called Y, was for a body color trait. The dominant alleles, big W and big Y, code for red eyes and a gray body. The dominant allele big W codes for red eyes, while the dominant allele big Y codes for a gray body. The recessive alleles, little w and little y, code for white eyes and a yellow body. In the experiment, they used individual flies with 2 different types of genotypes on their X chromosome. The females had 2 X chromosomes, one with the little w, big Y alleles, and another with the big W, little y alleles. These females were mated with males who possessed a Y chromosome, as all males do, and an X chromosome with a little w, big Y allele. I should point out that the Y chromosome is just the male sex chromosome. It doesn't have anything to do with the big or little Y alleles in this example. After these flies mated, they produced an F1 generation. Morgan looked at the genotype of the F1 generation to see if the allele combinations on any X chromosomes had changed. If each gene was tightly linked, one would expect the offspring to only have big W little y or little w big Y allele combinations. However, if there was a degree of recombination within the chromosomes, it's possible that the alleles would get shuffled around. The offspring would then have some X chromosomes that had little w little y or big W big Y allele combinations. What Morgan found was that the large majority of the F1 offspring had the same allele combinations as their parents, but he also found a minority of the offspring that had an X chromosome hosting little w little y or big W big Y allele combinations. He called these offspring recombinant, as they were allele recombinations different from those in their parents. The natural conclusion to this experiment was that genes on the same chromosome don't always stay together when they get sorted into gametes. This leads us to ask several questions. When I say they don't always stay together, what does that mean? How often do the genes not stay together? How does that process work? This process, by which genes can be shuffled around, is called crossing over. During a stage in meiosis, the homologous chromosomes swap segments of their DNA strands with one another. As homologous chromosomes carry copies of many genes, they only vary in the specific alleles at each gene locus. So when 2 homologous chromosomes cross over and swap segments of their DNA, the chromosome isn't being radically changed with new genes. It's keeping the same genes, but it's swapping out the alleles for those on the other chromosome. Genes that are located on the sex chromosomes, the X and Y chromosomes, are said to be sex-linked. Genes that exist on the Y chromosome will only be expressed in males, as only males have the Y chromosome. Genes that exist on the X chromosome can exist in both males and females, as both have at least one X chromosome. Males have the Y chromosome, while females have another X chromosome. So males have XY, females have XX. Recessive alleles on the X chromosome will be expressed more in males than females, as males don't have another X chromosome with a possible dominant allele to override it. Think of it this way. Each cell in your body possesses 46 deoxyribonucleic acid molecules, 23 chromosomes, or one copy of the genome, from your father, and another 23 chromosomes, or a second copy of the genome, from your mother. Every cell in your body has these 23 pairs of chromosomes. Chromosome pairs are numbered, as they contain genes for the same traits from person to person. For example, every human being has a gene that codes for the protein titin on chromosome 2. Titin is a small, spongy molecule in the muscle tissue that gives it a degree of elasticity. The gene that codes for a chloride channel protein called CFTR sits on chromosome 7. An error in chromosome 4 involving the uncontrolled replication of CAG nucleotide sequences causes Huntington's disease. The point I'm trying to make is that all human genotypes are roughly analogous. The same genes for the same traits are in the same places on the same chromosomes in the nucleus of the cell. Each set of 23 chromosomes can be paired with their analogous chromosome in the other set. These pairs are the homologous chromosomes. During a stage in meiosis, these homologous chromosomes are lined up close such that they can swap out segments of their mass with each other. In this way, alleles that come from your father may be transplanted onto the chromosome copy that came from your mother. Necessarily, your mother's alleles for the same genes would also have been transplanted from her chromosome copy onto the copy that came from your father. This is where the locus position on the chromosome becomes important. You see, the closer two loci are to one another, the more likely it is that they'll stick together and be sorted together. In this way, they're linked. There's a linkage between these two loci. But loci that are far apart from one another don't share this linkage. If the distance between them is great enough, it's quite likely that a recombination event will shuffle them around, or make them recombinant. We've created genetic maps of the chromosome, where we've literally mapped out the loci, where on the chromosome specific gene sequences are located. In fact, we can use the distance between two loci in a chromosome, measured in nanometers, to determine the percent frequency of crossing over events that happen between them. Generally, the percent frequency of a particular loci crossing over from one homologous chromosome to the other increases the further from the center of the chromosome it is. So the closer a loci is to the tip of an arm on the chromosome, the more likely it is to get swapped out during a recombination event. And this makes sense, as recombination events swap all the DNA past a specific site of action. It should be explained that not all genes operate on such a discrete level such as that in my examples. Not all traits are coded for by a single gene, or a proximal group of genes. These we call discrete traits. Some traits, on the other hand, are coded for by a large number of genes, which are scattered across multiple chromosomes. These are called quantitative traits. Human height, for example, is a famous quantitative trait determined by many different genes on many different chromosomes, as well as environmental influence like diet and stress. Quantitative traits are passed on through a form of phylogenetic inheritance, where each gene contributes a little bit towards the quality, like height. The more genes you have that contribute towards height, the taller you'll be, and vice versa. So I'm not quite done yet. There's still a little more detail I need to squeeze out of the basics of this lesson. I've talked about genes being dominant and recessive, where heterozygotes express one allele over another. But not all genes are so easily labeled. Some genes are codominant. This means that two different alleles for the same trait are simultaneously expressed. This is the case for the AB blood type, where the individual simultaneously expresses alleles for the A and B sugars that coat the surface of our red blood cells. Additionally, some genes show a kind of incomplete dominance. In this circumstance, the two alleles are expressed alternatively. Some cells express one allele, while other cells express the other allele. This creates a mosaic pattern among the cells, which appears to us and our giant macroscopic eyeballs as a mixed or blended effect. For example, consider the genes that code for pigments in human skin, or in the petals of a flower. In humans, if a parent couple is biracial and possesses significantly different skin tones, their offspring will receive genes for these different skin tones from each parent. Some skin cells will express the high melanin concentration of the darker parent, while other skin cells will express the low melanin concentration of the paler parent. When seen from our macroscopic perspective, their skin tone appears to be a roughly equal mixture of mother and father. The same can apply to flowers. Some species of flower can have white or pink petals. This color is due to the presence, or lack thereof, of a particular pigment in the cell. This pigment is expressed in the pink flowers, but it's not expressed in the white flowers. So if a plant with pink flowers reproduces with a plant with white flowers, the offspring's flowers will have light or pale pink petals. Some of the cells in the offspring's flower petals express the pigment, and other cells don't express any pigment at all. On a cellular level, this looks like a mosaic or a patchwork quilt. You've got some pink cells here, some white cells here. They're all scattered about. It looks like two different colors of rice all thrown together in a big pile. But when you look at it on a macroscopic level, when you zoom out to look at things on the Newtonian scale that we're used to, this mosaic blends together and you see it as a shade of light pink. Furthermore, the genotype doesn't strictly determine the phenotype. The environment plays a huge role in the expressed phenotype of an individual. Consider, for example, a gene that breaks down a particular biomolecule. If you eat foods rich in this biomolecule, it'll enter your system. If you have the gene, and it works, you'll produce the proteins that can break down and digest the biomolecule. If your gene is faulty or mutated, however, and it doesn't work, you won't be able to make that protein. Your body isn't able to break down the biomolecule on a chemical level. So if you eat it, instead of getting digested, it'll accumulate in your tissues and it'll cause problems like gastrointestinal distress, or in the case of a genetic disease like PKU, it'll cause neurodegeneration. By adjusting your environment, in this case by changing your diet to eat foods with low levels of the biomolecule, you can prevent these problems from happening. Alright, well, that's about it for this podcast. There's so much more information I could present, but I'm already going on longer than most episodes. There's so much detail here, and there's so much to explore and explain. I almost don't know where to start. I don't know what to cover because there's just so much. So I hope you enjoyed this podcast, and I hope you learned something cool. In the next episode, I'll be discussing the process of meiosis in great detail. As always, thanks for listening. If you enjoyed this episode, hit the like button and subscribe. Share the episode with a teacher, a friend, or a classmate. If you want to support the show, follow us on social media and consider buying something from the official store. Follow us on Twitter. Our username is @BioLogicPodcast. To buy something from the store, go to redbubble.com and search for BioLogic. Or go directly to the store at redbubble.com/people/biologic/shop. Become an official patron of the show. Sign up at patreon.com/biologicpodcast. Your support really helps the show, so if you like it, send us some love. And as always, thanks for listening.